All right, we are recording. So over to the slides. We do go and shoot. It's one of these. One of those. Yes, it is one of those. Great. Okay. So I've covered protists, and we've covered the early branches off the animal tree of life, sponges, cnidarians, a bit about placozoans and ctenophores. And for the record, I guess it's going to have to be tomorrow. I'm going to distribute a take-home exam, and this is as far as it's going to cover. So whatever's on there is going to be on protists, sponges, cnidarians, ctenophores, and why not placozoans. But we might as well start moving on and talking about all of the rest of the animal kingdom, which is in a super duper phylum. You know, we're kind of above the phylum rank here, and there's no really standard rank scheme. So we can call it a super duper phylum called the Bilateria. Animals with bilateral symmetry that have a defined left side and a defined right side. Whoops. Okay. This is sometimes lost or obscured. One example is a phylum called the Echinodermata, which we'll be talking about very shortly. Echinoderms include things like starfish and sand dollars, and many of them are radially symmetrical. However, we put them in the Bilateria because they evolved from bilaterian ancestors. Just because they have stopped having bilateral bodies as adults doesn't take them out of the Bilateria. You know, just the same way that the fact that I don't actually drive a wagon does not take me out of the family called wagoner, because that depends on my ancestry, not on what I actually do. So in some cases, this might be secondarily lost, but it's still there. Echinoderms, incidentally, develop from bilaterian embryos, which is in part evidence that the Bilateria is where they belong. Bilaterian bodies are made up of three basic cell layers that show up very early in development from a fertilized egg. An outer layer called the ectoderm, an inner layer called the endoderm, and a layer in between called the mesoderm. All three of these are true tissues. The cells are on basement laminas and fully connected together. And they appear very early in development and then fold and do various things to give rise to the full set of organs. And in Bilateria, we can finally talk about true organs. Something like a cnidarian might have specialized patches of tissue, like the gonads, for example, but it doesn't really have discrete structures that are made up of multiple tissue types integrated together. You know, the way your heart has muscle, but also has epithelial tissue on the inside and connective tissue holding it together, and nervous tissue that regulate the beat. Your heart's an organ. We haven't really seen that kind of integration in cnidarians before, but it's something that bilaterians have. So let's look at why some critters are radial. Here you've got a bunch of coral polyps, and their oral ends, the end with the mouth on it, is waving up in the breeze, or at least waving up in the current. And of course, you know, prey, little shrimp or whatever this thing is feeding on, is being brought in by the current and then, touching the tentacles and getting eaten. The aboral end is the end away from the mouth, and that is, of course, down below, attached to something. And the overall symmetry is radial. You'd see it if you went looking down on the mouth end. And it's correlated with the presence of only one gradient in the niche. What's a gradient? It's any kind of variable, any kind of environmental variable that varies fairly smoothly. So, for example, if you could take a little flow meter, something to measure the speed of flow, you'd find that it's very low when you're close to the bottom, and it gets faster when you go up through the water column. So the tentacles are being exposed to a lot faster flow than the base of these polyps is. And that means that, there's other gradients that get set up. The tentacles are probably exposed to more oxygen than the base is. The tentacles are exposed to more physical force than the base is. If it's not clear what I'm talking about, try going outside on a windy day. You feel the force of the wind on your face. You feel a lot less if you lie down. And that's because currents of air act just like currents of water in that they're slowed when they're next to a surface. So you feel less wind force when you're low than when you're tall. And it works with you on a beach, and it works with these polyps right here. The main thing that's determining how they're going to live is that flow gradient in the water column, with faster flow hitting the tentacles and slower flow hitting the base. Other than that, food or predators or currents, currents bringing oxygen or toxins or whatever it is they bring, all of those can come from any direction. So here in this picture, you see that's another coral polyp. It's radially symmetrical, and you can tell by the direction that the tentacles are pointing that the flow is coming from the bottom of the picture and moving towards the top of the picture, right? Because you can see the tentacles all streaming in that direction. But the next minute, the flow might change and be coming in from the top, or from the left, or from a bearing, I don't know, south by southwest 207 degrees. Whatever. That flow could be coming from any direction of the compass. And that means that anything that polyp has to respond to, whether it's food or a predator or a toxin, whatever it might be, could come from any direction. That polyp is going to have to intercept and respond to stimulus coming from any point on the compass. And that's correlated with radial symmetry. If you have this kind of lifestyle where anything could come from any direction, then you put the organs that you use to respond with out in all directions. You put your tentacles out in all directions, surrounding the mouth equally. And that's radial symmetry. Bilaterians, which is everybody that isn't a cnidarian, ctenophore, placozoan, or sponge, are often adapted for environments in which everything is going to come primarily from one direction. Now, this critter is a cnidarian. It's a sea pen. It's one of these colonial soft corals, one of these colonial octocorals. And the individual polyps, which in this picture are really tiny, the individual polyps are radially symmetrical. They're perfectly good little octocoral polyps. However, the entire body of the whole colony is bilaterally symmetrical. You can see that there is a central main polyp, and then you have, polyp leaves on the left side and on the right side. And remember that bilateral symmetry means that there is only one plane that you could divide something in, or that you could divide something along to make it fall into equal mirror image halves, right? There's only one way that I could chop you in two, not that I would, that would split your body into halves that were the same shape but mirror images of each other. It's the same thing here. There's only one way you could cut the C-pen to divide it into equal mirror image halves. The C-pen has a left side and a right side. And the reason it does is these guys tend to live in tidal channels, channels in relatively shallow water that feel the force of the tides coming in and going out twice a day. What that means is that it's pretty predictable in this environment where the flow is going to be coming from. It's going to be, the tide is going to move in one direction down the channel, and then it's go back out in the opposite direction. And then it's going to move in, and then it's going to come out, and all of the time it's going to be either coming from one direction or coming from the exact opposite direction. So you have bilateral symmetry correlated with an environment in which everything that the organism is facing, right, all of the predators, all of the prey, all of the stimuli, all of the currents are going to be coming in primarily a single direction. So you get bilateral symmetry correlated with that kind of regime. And you even get it in things like this. On the left, that's a cell phone tower. And of course, you've got plenty of cell phone towers that you can see around you. And the cell phone tower is radially symmetrical. It would look radially symmetrical if you could stand at the base and look straight up. On the other hand, that radar antenna is bilaterally symmetrical. If you were to stand at the base and look up, you could define it as having a left side and a right side, whereas a radially symmetrical cell phone tower does not have a defined left and right. And the reason is the same as for the C-pen. The cell phone tower picks up signals coming in from any direction, right? Your cell phone could be on any compass bearing relative to the tower, so the cell phone tower has to pick it up from anywhere. The radar antenna detects signals only from one direction. So again, if things are coming from your environment primarily from one direction, it's advantageous to be bilateral and to put all of your sense organs and responses out in front to meet that signal. Whereas if things are coming from any direction, it's advantageous to be radially symmetrical. And most bilaterally symmetrical organisms are motile, they move. And that means that most of what they face is going to come from one main direction, and that direction is dead ahead. This happens to be a swimming deep-sea segmented worm, probably genus Tomopterus, I'm not sure. And it's going to spend its life swimming through the deep ocean. And most of the things that it has to respond to are things that are going to come from ahead, things that it's going to be moving towards, things that it's going to, probably not literally, but things that it's going to run into. So bilateral symmetry is often correlated with an active lifestyle, because if you're moving forward, that creates a gradient in which stimuli tend to come from one primary direction. And bilateral symmetry is often correlated with cephalization. This is a very weird critter, actually fairly common in marine plankton, but not the sort of thing that you're likely to see unless you actually go out looking for them. These are some small planktonic worm-like invertebrates called chaetognaths. There's a complete one over on the left, and on the right, that's a scanning electron micrograph of the head of one of them. and it looks really weird. Those hook-shaped bristles are actually used to trap prey and shove it into the mouth. It's not familiar to you, but you can still see cephalization on it because those feeding bristles and the mouth and sense organs and also the brain are all shoved into the head end, shoved into the anterior end, simply because if you're moving forward, the things that you have to deal with and respond to are going to be coming from ahead, right? It's the same reason that the windshield and the headlights and the air intake are on the front of your car and not the back of your car, right? Because your car is mostly going to be meeting things, face on. If you don't believe me, drive through a swarm of bugs, and you will notice that your car encounters a lot of things head on. And so the structures that you need in order to meet things that are coming at you from head on, like headlights and air intakes, go on the front of your car. And it's the same thing with organisms. So bilaterally symmetrical organisms tend to have cephalization, sense organs and neural processing organs, i.e. a brain, tend to move to the front to be ready to meet whatever the organism faces. And we have a lot of organisms that are members of the Bilateria by descent, but they've evolved radial symmetry if they've got a lifestyle in which stimuli come from any direction. This is a diagram of a segmented worm, a distant cousin to earthworms. It's marine. It's called a feather duster worm, and lives in the ocean and constructs a papery tube surrounding its body. You've got the tube on the right. On the left, that's the worm removed from its tube. And the body is a little twisted here, but it is bilaterally symmetrical. You can see little appendages coming out, and the worm does have, each segment has a left appendage and a right appendage. So the worm body is still bilateral, but the way it lives is it spends all of its time in that tube. It can't survive without it. And it sticks a whole bunch of tentacle-like feeding organs called radioles out of the tube. So the actual body of the worm is deep in that tube, and the only part that comes into contact with the outside is that bundle of branched tentacle-like radioles. And that bunch of radioles is basically radially symmetrical. This is what one of these worms looks like. You can find, they sometimes have these at that pet store called Pet Country. If you don't make a special trip, but if you ever happen to be there, there's a number of segmented worms that are sold under the name feather duster worm or sometimes peacock worm that have a similar lifestyle to this. And although they're classified in the Bilateria, the part of the body that they actually stick out and use to filter feed is radially symmetrical or very close to it. We'll see some of those probably next week. The same is true of living echinoderms, most of them. This is a critter called a basket star, similar to a type of starfish called a brittle star, but the arms are very, very highly branched. So you don't have 5 nice, neat little arms. You have arms that branch and branch and branch again. This guy is radially symmetrical, but Astracme, this brittle star, feeds by climbing up on something, be it a rock or a soft coral or whatever it is, and extending all of those arms out into a big filter feeding basket. Just letting all of those highly branched arms wave in the breeze, trap food particles, and pass them down to the mouth. So that, again, is the kind of lifestyle where things can come at you from any direction. And because of that, the anatomy has evolved to a radial symmetry, even though by ancestry, ostracme is in the bilateria, and it evolved from bilateral ancestors. So, yeah, you can actually see several cases where a lineage has shifted from being, a filter feeder to an active mover, and that usually correlates with a shift from bilateral to radial symmetry, or vice versa, right? Bilaterian bodies usually work like this. Remember that muscles can only contract. A muscle can't stretch. To stretch a muscle, what you need is another muscle pulling in the opposite direction. In you, you have opposing muscles, typically on opposite sides of bones. If you contract your biceps, right, you bend your arm, and you probably can't see this on the camera, but you contract your biceps and bend your elbow. If you want to straighten your elbow, you relax your biceps and you contract a muscle on the opposite side of your elbow, your triceps muscle. And it's the same thing for the other muscles in your body. Your quadriceps, flex your thigh in one direction, but then your hamstrings flex it in the other. I might have that backwards, but the basic idea is that you have muscles that tend to come in pairs that counteract each other, such that when one contracts, the other is stretched. In lots and lots of bilaterians, we're going to see this in several times in different groups. If there isn't a skeleton that muscles attach to, as there is in vertebrates, you'll very often have circular muscles that are shaped like circles, and they work against longitudinal muscles, which are muscles that run along the long axis of the organism. So when the circular muscles are contracted, the organism becomes narrow, and the longitudinal muscles are stretched out. The longitudinal muscles are stretched, and the organism becomes very long. When the longitudinal muscles contract, the organism gets a lot shorter. It also gets a lot thicker, and the circular muscles are stretched. And very often that's associated with a body that's filled with a liquid-filled cavity, which is called a hydrostatic skeleton. It's not a bony skeleton, and it works because water is, for our purposes, incompressible. You can't pack water down into smaller volumes, at least not by the kinds of forces that living things experience. Even the extremely high pressure at the bottom of the ocean does not affect the density of that water very much. Affects it a little bit, but much less than you would think. Even water that's being pushed on by 6 miles of water above it is not significantly more dense. It's not compressed very much. And certainly the muscles of a worm can't compress water at all. So a lot of bilaterians work like, water balloons. Think about playing with a water balloon, and, if you squish it, the water can't be packed down. The water always maintains the same volume. So if you squish a water balloon to be narrow, it gets longer. And if you compress a water balloon to be short, it gets thick. And of course, if you squeeze one part of a water balloon the part that you're not squeezing gets larger and longer. But in all of those cases, the volume of the fluid inside doesn't change. You can't compress water just by squishing it. And so animals with a hydrostatic skeleton can burrow this way if they relax some muscles and contract others in an orderly cycle. So if you look on the over at A on the left, that's a worm. What it might do is at the back end, it will relax the circular muscles and make that part of its body wider so that it pushes against the substrate. But okay, let me think. Okay, by relaxing longitudinal muscle, it'll relax circular muscles and contract longitudinal muscles at the back end, which makes its back end stubby and thick, so that pushes against the sediment and holds the worm in place. That's called a penetration anchor. At the front end of the worm, the circular muscles contract, and that forces the head of the worm ahead. So the worm pushes its body, the front end of the body, through the sediment, while the back end is being held in place by that penetration anchor. And then if you reverse that and relax the circular muscles at the head end will swell, and that holds the head end of the worm in place, while contraction of the longitudinal muscles will pull the tail end of the worm forward. And then you just keep repeating this cycle where you alternately inflate the tail and push the head forward, or inflate the head and pull the tail in. And repeating that cycle enables the worm to burrow. And it works because the worm can relax some of its muscles while contracting others, always maintaining a constant fluid volume. Gods, I hope that made sense. When you've got an animal that's not only a fluid-filled bag, but is segmented as well, you can do even more sophisticated things with burrowing, because each segment can act as an independent hydrostat. So some of these segments, like up at the top, segment 9 is very short but very wide, and so it can form part of the penetration anchor that's holding the head of the worm in place, while segment 3 up at the top is elongated and narrow and can push forward. And then in the picture below, you see that segment 3 has gotten shorter and thicker and is now pushing against the soil and holding the head of the worm in place. So every segment cycles from being short and stubby and sticking in place to being long and thin and being pushed forward. And cycles of that, where each segment takes turns being one or the other, is fundamental to the way that earthworms burrow. Okay. In some bilaterians, there's no open cavity in the body other than the gut. You see this a lot in some very small bilaterians that aren't big enough to burrow in the first place. And bilaterians like that are said to be acoelomate. A coelom is a fluid-filled body cavity. And if you don't have one, you are acoelomate. But in a great many bilaterians, there is a large fluid-filled cavity in which the gut is suspended, and it's lined with a specialized type of epithelium. Remember, the tissue that lines cavities is epithelium. That specialized epithelium is called mesoepithelium. So if you've got a big fluid-filled body cavity that's lined with mesoepithelium, that's called the coelom. And to give you some reference, you've got a coelom, your coelomates. Your coelom is divided into 3 different spaces: the pericardial cavity, is the open space around your heart. The pleural cavity is the space around your lungs, and the peritoneal cavity is the space around your guts. Your intestines are in your peritoneal cavity, and mesoepithelium makes up structures called the mesenteries, this kind of tissue webbing that holds your guts in place. You don't have a great deal of fluid sloshing around in that peritoneal cavity. At least hopefully you don't. If you do, it's a medical condition called dropsy, which can actually mean several things. It's the kind of thing that people get with, I think it's portal hypertension, high blood pressure in the liver, which can result from alcoholic damage to the liver or something like that. But yeah, you don't normally have a lot of fluid in your peritoneal cavity. So the cavity has, doesn't have a lot of space normally, but it is still in fact there. One of the effects of that is that it isolates the muscular contractions of your intestine from the muscular contractions of your body, meaning that your intestines can push the food on through unaffected by what the rest of your body is doing. You don't, automatically, squeeze food or waste through your intestine just by bending over, or, just by breathing for that matter. Your intestine can do what it does, and it's insulated from the movements that your body wall is making. And the same thing with the others. You can't compress your heart just by bending in a funny way. The pericardial cavity gives your heart room to work, and the rest of your body is isolated from the heartbeat. So anyway, in some very tiny bilaterians, this one happens to be a microscopic bilaterian that belongs to a group called the gastratrix, which we may or may not see. There may not be any need for a coelom at all. These are, millimeter size, maybe not even that much, and every cell can get nutrients and oxygen purely by simple diffusion. So there's no real need for a coelom. There are some large acoelomates like this one. This is a marine flatworm related to the planaria worms that you might remember. It's called aquaplana. And the white branchy stuff that you see in the center of the body is the main gut. And that gut is very highly branched, and you can see smaller, finer, kind of grayish branches, branching and rebranching, almost looking like moss, extending through all parts of the body. Those are branches of the gut. The gut is so highly branched that it can essentially act like a circulatory system. All parts of the body can get nutrients through these gut branches. The worm is probably so thin that all parts of the body can get oxygen just by diffusion. But then in coelomates, here's a cross-section through an earthworm. The brown crescent-shaped thing in the center is the gut. There's a blood vessel on top of the gut. That's the red thing that you're seeing in cross-section. Below the gut in bright green, you've got a nerve cord and branches of it. And then the body wall is made up of muscles, which are colored salmon pink in this case. And then there's that open space in between that's not colored in at all. That's the earthworm's coelom. The coelom is filled with fluid. In this one, it's actually pretty spacious. And that thing in the coelom that looks a little bit like a tuba designed by Dr. Seuss is called a protonephridium. That's an excretory organ. Earthworms get rid of waste by filtering the coelomic fluid, and that funny-looking tubular thing drawn in very light blue is a protonephridium that filters waste out of the coelomic fluid and expels it out of the body through a little pore that you can just see. So in this earthworm, the coelom is very spacious, wide open, and earthworms use it because they burrow using those hydrostatic skeletons. The hydrostatic skeleton in this case is the coelom. If you're wondering about the difference, a hydrostatic skeleton is defined on how it's used. A coelom is defined on what structure it has. There are burrowing sea anemones. I actually have seen some in Baja California, Mexico, that can burrow by sealing the mouth and just using their fluid-filled gut as a hydrostatic skeleton. So that's not a coelom, because sea anemones are not bilaterians, but it's functioning as a hydrostatic skeleton. So hydrostatic skeletons are called that because of what they do. Coeloms are called that because of what they are. Many bilaterians have a separate fluid-filled cavity or set of cavities filled with a fluid that's specialized for transporting oxygen, in other words, blood. A fluid-filled system that's filled with oxygen-transporting liquid is called a hemocoel. All right, hemo for blood. There are cases where a hemocoel can act as a hydrostatic skeleton. All right, I'm trying to think of an obvious example that might not be potentially bothersome. Oh, what the heck, we're all grown-ups here. The erectile tissues of the penis are an example of a hydrostatic skeleton that's connected to the hemocoel. It's blood flowing into those tissues that gives the organ the particular rigidity of which some of you may have heard. Right. The erectile tissues of the clitoris function in the same way, by the way. Let's be equal opportunity here. Those are both examples of a temporary hydrostatic skeleton that is part of the hemocoel. Right. Sometimes the coelom has a role in gas exchange as well as being a hydrostatic skeleton. And the general rule in invertebrates is if the hemocoel is big, the coelom is small. And if the coelom is big, the hemocoel is small. One or the other is going to be predominant, but usually not both. So for example, that's a rough diagram of a snail. And by the way, you really ought to thank me that I'm not making you guys dissect a snail in this class, because snail anatomy is weird. Partly because the body undergoes this twisting in early development so that the anus is on top of the head and the gut forms a loop. And on top of that, you have the coiling of the body to accommodate the shell in many snails. So yeah, you've got a coiled and twisted body right here, and you certainly don't need to know all of the details there. But the hemocoel is large, and it's got vessels, and it's got a heart, but it's also, the vessels connect up a whole bunch of open spaces called blood sinuses, in which the blood just kind of sloshes around. In what we call the crawling foot, the thing that the snail actually creeps around in, blood spaces in the foot can actually act as a hydrostatic skeleton. You see this in burrowing snails. You definitely see it in burrowing clams. The muscular foot of the clam can be used to burrow, very much like what we've already seen, because it's filled with these open blood spaces with blood sloshing around. There's a coelom here. The actual coelom is those small spaces around the kidneys. labeled AK and PK, those receive and they expel urine. And the only part where you get a real coelom is just those little spaces around the kidneys. So in snails, the hemocoel is big and the coelom is small. In you, the coelom is pretty small and the hemocoel is quite large. In starfish, that would actually be a good counterexample. The hemocoel is small. There is a circulatory system, but it doesn't amount to all that much. And the coelom is large. So usually one is big and the other is small, but usually not both are well developed. Hello, cat. Okay, tell you what, I'm being informed that, yes, this poor cat has never been fed in her entire life and is desperately starving to death. So I'm going to go ahead and say hello to the nice students, Yelling Goat. Yeah, this is Yelling Goat. Hi, kitty. Good kitty. I'm going to go ahead and stop recording for about 10 minutes and go feed the cats. We'll pick this up at, I've got 9:56. Let's pick this up at, what the heck, 10:10. We'll go on for about another 45 minutes. That'll be it for the day. And this afternoon and tomorrow, I finally am going to be able to get caught up on the labs. So yeah, I will see you folks at 10:10, as they used to say on CB radio, 10:10 till we do it again.